Okay, we are continuing with biochemistry in carbohydrates. We started earlier looking at monosaccharides, which are the basic units of carbohydrates. We did some classification of these monosaccharides based on the functional group that is present in their structure. If it is aldehyde, then we classify those monosaccharides as aldoses. If it is a ketone, we classified those monosaccharides as ketoses. We also classify them based on the number of carbons that are present in their structure. So we had the smallest being trioses, containing 3 carbons. We have tetroses, pentoses, heptoses, all the way up to, I think, 9 carbons, nonoses. But the most common that you come across are the pentoses and hexoses. And I mentioned that trioses themselves are not carbohydrates, but in the metabolic pathway of carbohydrates, these carbohydrates break down to the smallest, which are the trioses, like glyceraldehyde, dihydroxyacetone, which will continue in a reaction path, to generate energy. Then we also looked at the naming of these, particularly the pentoses and the hexoses, with their ending in a suffix ose, ose. So we looked at the fact that many of these monosaccharides, even disaccharides, you realize that the name is ending with ose. So like glucose, sucrose, galactose, lactose. So we saw that. Then we went ahead and looked at stereoisomerism of these monosaccharide, looking at their differences in orientation in space, so that you can have different monosaccharides that are having the same molecular formula, but are having different structures. And under this stereoisomerism, we saw 2 kinds of stereoisomers. We can have stereoisomers that are mirror images of each other, and these we refer to as enantiomers. And there are some stereoisomers that are not mirror images. These are diastereomers. We even looked at what we called epimers. So 2 monosaccharide molecules that are having a difference in their structure only around a single carbon. And then last week, we continued to look at cyclic forms of the monosaccharide structures, which we referred to as the Haworth projections or Haworth perspective formulas. And we compared that to the straight chain structures, which we were looking at earlier, which were the Fisher projections. We referred to the first carbon in that cyclization where you can have a rotation of the groups around it as an anomeric carbon. And so the isomers of these cyclic forms, we refer to them as anomers. So these were the things we did previously, a lot of things about carbohydrates. We are continuing today to look at modifications of monosaccharides. You can say monosaccharide modifications. So that's what we'll do today, modifications. And then when we meet next time, it will be left with the very last thing here, which will be the oligo and polysaccharides. So then we will then start to form polymers. So we'll now be putting the monosaccharides together and see the kind of large structures that can be formed from these basic units that we are referring to as monosaccharides. So let's continue and we'll look at these modifications. What I intend to do is there are so many monosaccharide modifications. We just cannot say we are covering all the modifications of monosaccharides. But what I tend to do is to introduce you to some of the very common monosaccharide modifications you come across as you continue to study biochemistry in second year, third year, fourth year. You'll be meeting some of these modifications. Some of them, you see them in the metabolism of carbohydrates. Some of them, you see them in the structure of some components in some other organisms like bacteria, fungi. You'll be hearing their names, so that by the end of this lecture, I hope that your interest will be triggered into these monosaccharide derivatives. And so you go and be looking at the different kinds, the ones that we will not even look at. And also when you read and you meet any of these, you can quickly relate with it and say, and know that this is a monosaccharide modification. So I hope that by the end, your interest will be that whipped up to get to know more about these modifications. Generally, monosaccharide modifications are compounds that that are derived from a parent monosaccharide. That if you see on the structure of these monosaccharides we have seen so far, I mentioned that they are all polyhydroxy, meaning that you have hydroxyl groups all over on the carbons that are in the structure of these monosaccharides. But in the monosaccharide modifications, you will see that in place of one or more hydroxyl groups, you have a different substituent, something else, some other. It could be some residue of a molecule that is replacing a hydroxyl group, an OH group on a carbon. So once you see that, that becomes a derivative or a modification of the parent monosaccharide. So what I mean by that is, so let's take the very first example that we have here, which is a glucose derivative. And this is glucose 6-phosphate. Glucose 6-phosphate. Now we see the glucose molecule is having a hydroxyl group on carbon number 6. So that is the carbon that is out of the ring. And you see CH2OH. So that is inside the glucose molecule. But you see this derivative, which is glucose 6-phosphate, is having, in place of the hydroxyl group on that carbon number 6, we have a phosphate group. So that is a derivative or a modification of the glucose molecule. And this glucose derivative you will come across in major metabolic pathways of carbohydrates. So if you go ahead and you're looking at the metabolism of carbohydrates and you look at the process of glycolysis, that is the breakdown of glucose. And remember, glucose is a hexose. It's having 6 carbons in it. In the process of glycolysis, the glucose is broken down into 2 molecules. Each of them will have 3 carbons in them. In that process, the beginning, that is the activation of the glucose molecule, is by phosphorylation, where you have a phosphate group from ATP onto glucose. So then you have this glucose 6-phosphate before other reactions occur all the way until that glucose molecule is broken down. By the end, you get some 2 molecules of pyruvate, which is a 3-carbon molecule. But you can also have even this glucose 6-phosphate not going down the path of that breakdown, but can also go into even storage. That is, if it was in plants, it goes into the synthesis of starch. So biosynthesis of starch, you have these glucose 6-phosphate molecules getting polymerized. So you have some synthesis of amylose, amylopectin, and all of this comes together as a storage in plants that can be stored in some roots for root tubers, stems for stem tubers, or even seeds. So like cereals, for instance. So energy is stored in these plants. And because we are not photosynthetic organisms, we cannot prepare carbohydrates by just taking carbon dioxide and water with energy from the sun to prepare carbohydrates. So we then go for the carbohydrates from these plants. But we also have a storage system in us where I think I mentioned it before, that excess carbohydrates in us, we store it in the form of glycogen in the liver and in the skeletal muscles. That process also, you have the glucose 6-phosphate, entering into that pathway to synthesize glycogen, which is stored in the liver. Or you also have the glucose 6-phosphate also going into another major metabolic pathway, which goes into the synthesis of nucleic acids, which is DNA, RNA. So you have the synthesis of the ribose 5-phosphates, which then will go into the synthesis of RNA. Deoxy, some you have some deoxygenation or deoxylation of such ribose 5-phosphate will now give you the deoxyribose phosphate, which will then go into the synthesis of DNA. So the glucose 6-phosphate is something you come across all over. Like if you are looking at major metabolic pathways of carbohydrates, you surely come across the glucose 6-phosphate. And even a lot of time in clinical settings, you even talk about some deficiency of an enzyme that is related to the oxidation of these glucose 6-phosphate, which is the glucose 6-phosphate dehydrogenase. You hear a lot about it when you continue. Now, another one is glucosamine. Glucosamine. So here too, you have the glucose molecule on the left, and you have glucosamine on the right, which you can compare to the glucose molecule. And you realize that on carbon number 2 of glucose, where you have a hydroxyl group, you see that hydroxyl group is substituted with an amine group in glucosamine. Now, glucosamine can serve as a precursor for the synthesis of many glycoproteins. These are complexes of carbohydrates and proteins. Of course, if you remember when we were doing proteins, peptide bonds will be forming between an amine group of an amino acid and the carboxylic acid group of another. And so if you have a glucosamine, it means that a protein can easily bind with this amine group, with a peptide-like bond, that is with the carboxylic acid group and the C terminus of the protein that is coming to bind onto glucosamine. So that is how come glucosamine can be a precursor for the synthesis of glycoproteins. You'll find glucosamine in local pharmacies, like you find it with different brand names because you have I intentionally have put a picture here of one of these glucosamine capsules that are in the pharmacies that is purchased as some food supplements, usually used for say joint pains, even though in some countries it's not approved as a prescription drug, but in Ghana, people with some joint issues, when they go to the hospital, glucosamine is prescribed for them, so they go to the pharmacies to get these glucosamine capsules, and it's used for such purpose, say for joint issues, because glucosamine plays some role in the synthesis of tendons and cartilages of the joints, and even the synthesis of the synovial fluid that you find between joints for lubrication of the joints, so that the use of it can enhance the production of of such lubrication at the joints. So yes, you hear of glucosamine in the pharmacies. Then even looking at glucosamine, you can have that uh, uh, modified further into what we refer to as N-acetylglucosamine. So here I've put the glucosamine here, what we looked at just in the previous slide. So you see glucosamine on carbon number 2, you have an amine group there. Then you see N-acetylglucosamine, where on that amine group on carbon number 2, you now have a residue of acetic acid being bound onto that amine group. So that is why it says N-acetyl. The N refers to the amine group there. You have the conjugation of the residue from the acetic acid on nitrogen, of that amine group. That's why you have at the beginning N. So N, the residue is it's an acetyl group. It's coming from acetic acid. So you have that N-acetylglucosamine. And now N-acetylglucosamine, you come across it in bacterial cell walls. So N-acetylglucosamine forms a polymer with another derivative, which we'll go to soon. And that gives you a polymer that gives a protective coating around bacteria. So that is you find it in some bacterial cell walls. We'll look at it further when we go to the next derivative, because that the N-acetylglucosamine forms what we call a call it a heteropolysaccharide. So a polysaccharide that is formed from different monosaccharides. That's a heteropolysaccharide. And so you will see the second derivative that will form the compound, which is the main component of the cell walls of bacteria. You will also find it as a major component of the exoskeleton of arthropods, like insects, crustaceans like crabs, lobsters. You'll find N-acetylglucosamine, this time around forming a homopolysaccharide. That's a polysaccharide that contains only N-acetylglucosamine. That polysaccharide is chitin, which is a major component of the exoskeleton of these organisms. We'll actually take a look at these polysaccharides much more in detail when we are looking at the oligo and polysaccharides. That is in our next lecture, when we start to look at bringing these monosaccharides and the monosaccharide derivatives together to form oligosaccharides and polysaccharides. Then we'll look at them and see the bonds that are between them and all of that. Now we move to, so this is the monosaccharide derivative that will be forming what we call peptidoglycan, which is the major component of bacterial cell walls. But even before then, let's pay attention to the differences, or in comparison to the glucose molecule, what makes this derivative. So first of all, we look at muramic acid, which is at the center, muramic acid, and we see Just like the glucosamine on carbon number 2, moramic acid is having the amine group there in place of a hydroxyl group. But in addition to that, on carbon number 3, there is another substituent which is depicted R. And that R is actually a residue from lactic acid. So that is a lactic acid residue. So we see that moramic acid, but moramic acid naturally it occurs as in its N-acetylated form as N-acetylmoramic acid. What you see at the left of your screen, you see that you have on the amine group of moramic acid, you have the conjugation of lactic, you have a residue of acetic acid on the amine group. So you have an N-acetylated form of the moramic acid. Now, this N-acetylmoramic acid is what binds with N-acetylglucosamine. So these two molecules alternate. So if you have N-acetylglucosamine, N-acetylmoramic acid binds. Then you have another N-acetylglucosamine binding to that. Then it continues. So alternate, they will be alternating in a chain. N-acetylglucosamine, N-acetylmoramic acid, N-acetylglucosamine, N-acetylmoramic acid, alternating like that to form a long chain structure, which is called peptidoglycan. And it is that peptidoglycan which is the major component of bacterial cell walls. So I said that we will look at when we are looking at the oligo and polysaccharides, we'll also look at the polymers that can be formed from these modifications. So then we can look at how that structure will look like, how that polymer will look like. So we move to another, which is also a glucose derivative that you also come across along the line. That is glucuronic acid. Glucuronic acid, sometimes you can also refer to it as a glucuronate. So if you see, when you are reading and you see glucuronate, it is the same as glucuronic acid. Now here, what you see is that a glucose molecule, everything is the same as in the glucose molecule, except on carbon number 6. So carbon number 6, you remember that was CH2OH, and that has been oxidized. So there has been some oxidation of that group, and you now have a carboxylic acid group in place of that CH2OH. Now that is an acidic sugar. In fact, we were actually all the like this moramic acid and all we were seeing above, they were acidic. Of course, it's in the name. This is one of such acidic sugars that we refer to as a uronic acid because it was first isolated from urine. Now, you see glucuronic acid in the metabolism of foreign substances, what we refer to as xenobiotics. If you was a substance with some pharmaceutical property, then we will be referring to say metabolism of drugs. But even if he wasn't having a therapeutic effect, but rather some toxic effects, like that is to say that it's a poison. All of these, either the thing is having a pharmaceutical or it's having a toxic effect, they are foreign substances that we refer to as xenobiotics that undergo some metabolism in the liver. So just like the way when you take in food, we talk about digestion. The food goes through your mouth, through esophagus to stomach, and is broken down. You refer to different enzymes that are working from the mouth, salivary amylase, and in the stomach, you talk about pepsin and all of that. And then in the small intestines, food gets absorbed, all that kind of thing. The same way, when you take a medicine, or if it's a toxic substance that enters into the body, the metabolism of that substance occurs in the liver. So it is the liver that um, acts on the substance. If it was a toxic substance, to detoxify it, make it less toxic to be excreted out of the body. Of course, sometimes some of such processes rather causes these substances to be more toxic. And one of the processes that you find in that metabolism of these xenobiotics, these foreign substances, is what we refer to as glucuronidation. So in that process, you have glucuronic acid binding onto the substance. So that is a process we refer to as conjugation. In that manner, what happens is that you have many of these, most of these drug substances are lipophilic, so that they are not going to be water soluble. And so in order for them to become soluble in water and be excreted mostly out of urine, glucuronic acid binds onto these substances so that they can be easily moved out of the body in aqueous media, mostly in the form of urine. So that process is what we refer to as glucuronidation. So glucuronic acid has such a major function in the metabolism of drugs in the body. Then we move to another acidic derivative. This time around, it is on carbon number 1 that you have the oxidation. So actually, this is a straight chain structure. It's just that it's folded. If you look at it, you realize that this is not a ring structure. It's just that it's folded so that you can easily relate with it when you are looking at it against the Haworth projections. But you realize that on the first carbon, which was supposed to be an aldehyde group. So if this was an aldehyde group, you have the C double bond O, and in place of this O minus, so this O minus is actually supposed to be OH if it was in its molecular form, but now it's in its ionic form, meaning that it has given out a hydrogen ion, because the carboxylic acid group can give out a hydrogen ion. In place of that OH group, you should be having a hydrogen. That is the original glucose molecule. There's a hydrogen there, so that you have an aldehyde group. So you see it's an oxidation of that aldehyde group to give you a carboxylic acid group there. And because of that, we refer to it as an aldonic acid. So aldonic coming from aldehyde. So it's the aldehyde group that was that was oxidized to give you that aldonic acid. This is naturally occurring in fruits, honey, wine. Of course, wine is brewed from some fruit, so you can have aldonic acid in there. This gluconic acid or gluconate, you can have it in such, and because of that, it means you can it's used as a natural food additive, for like an acidifier to regulate acidity of foods. Another use you come across in veterinary medicine, this gluconic acid or gluconate is used in combination with the amino acid arginine, but in the form of the salt of zinc metal, so that you have what you refer to as zinc gluconate injection, which is used for chemical castration of male dogs. So if it's injected directly into the testes of these male dogs, and that results into neutering, or say what you refer to as a chemical castration of these male dogs. So it has such veterinary use as well, apart from it being used as a food additive, regulating acidity in foods. You can use it in the form of its salt with zinc and the amino acid arginine for that veterinary use. Right. So we move on to another, which is gluconolactone. So you can see again on the first carbon that what was the original anomeric carbon, the anomeric carbon of glucose. You see there is a double bond O. That's like a cyclic ketone that is formed over there. It's also an acidic, because it's an aldehyde group that has been oxidized to a ketone group. So you can come across this also as a food additive, used as, say, a sequestrant. Okay, another way you can refer to a sequestrant is, say, a stabilizer. So stabilizer in foods like foods with lipids, or say fats and oils in them. You find gluconolactone used as a stabilizer, which will be preventing the oxidation of free fatty acids, so that you don't have these oils, turning rancid, then it begins to give some sort of a rusty smell of the food and even the taste of it. So you find that gluconolactone is used sometimes. It's like a chelating agent that will bind to some metals like copper, zinc, and because these metals, they also act as some cofactors, to lipases that can result into the breakdown of these free fatty acids, resulting into their oxidation, and then you start to have such unwholesomeness of food, the taste of it, even the smell of it, sort of make it not pleasant. At the same time, it can also be used as an acidifier, because it's an acidic derivative, so it can also be used as an acidifier, sort of controlling pH in food substances, even as a leavening. In fact, we in Ghana are very much used to using salt for leavening. That is preservation of food. I mean, what actually that does is, if you add salt to, for instance, fish or meat, it absorbs water from the meat or the fish or whatever it is you are preserving. It absorbs, or maybe even if it is condo or anything that you are adding salt to, the purpose of adding the salt is for the salt to bind to water so that you don't have free water in the food substance. Because when you have free water up to a certain percentage, then you start to have the growth of fungi in the food substance, like some molds will start to grow, and that will definitely cause food spoilage. And for even liquid foods, for instance, those ones will even have conditions that are ripe for the growth of bacteria in such foods. So when you're adding salt to this substance, what you are doing is actually what you're doing there is curing. That is curing. Leavening is what you do with kneading of wheat dough. So you sort of use a mechanical force onto the dough just to soften it and all of that, so that after you add some yeast, you put it down, it can, when you can easily bloat, it can easily the dough will sort of rise because of metabolic processes of yeast on the wheat dough, which will produce carbon dioxide to let the dough rise up. That is leavening. But you can actually achieve that also with this natural substance, which is gluconolactone. So industrially, this will be much more practical than the mechanical way of doing it. And what it does is that this gluconolate has it will cause some sort of foaming action, which will then result into the rise of the dough in the bakery. Aside using the gluconolactone also as for curing, what I was referring to earlier. In our jurisdiction, we use salt a lot for curing, but in some jurisdictions, when you buy some meat, fish, sometimes the taste is sweet. See if it is sugar that is used for these things. It is actually some of these substances like gluconolactone that are used for curing of the food substances. And we also have what we refer to as pickling. You see this usually in countries that are in the temperate regions, having very long winters, so that when it is like summer and they grow some vegetables and fruits and they want to keep it for a very long time, they can put this like say cucumber or some you have some of these fruits like berries and things like that they want to pickle. They put it in a jar, some big jar, and then they add say either sugar or salt. Like they form a brine. The brine is some salt solution they add to it, and then they can cover it, seal it, so that oxygen cannot get there. So that thing can be kept for a very long time, and anytime that they want to eat, they just go pick one of the jars, open it, and remove this out of it. That is some way of preservation of these food substances. Once you have salt in there, that will absorb moisture out of these food substances. The foods don't get spoiled. They can be kept for a very long time and be eaten. So that is pickling, and you can have this gluconolactone also used in that process of pickling of these food substances. Well, let's move on to what we refer to as an amino sugar. So, of course, we have seen some amino sugars already, referring to glucosamine. So glucosamine is an amino sugar, but that was specifically a derivative of glucose. But here we're looking at another example. It can be with another monosaccharide, which is galactose. So we see on carbon number 2 of galactose, you have the hydroxyl group replaced with an NH2. So that gives you galactosamine. Now, what you should pay attention to here, don't get confused when you see some of these structures that the hydrogens are not written. They are the same. It's known that there is hydrogen there. So even if the hydrogen is not written there, it is known that it's there. So you see on the right side, yes, the hydrogens are written, but on the left, in the galactose molecule itself, you don't have hydrogen written there. So don't get confused about that. It's the same thing that is being shown. You can choose to write it, you can choose not to write it. It is known that on each of the carbons, there will be a hydrogen in addition to any other group that is there. And usually you will want to show the hydroxyl group because the direction in which it shows is important for a particular monosaccharide. So we see that, yes, we can have galactosamine as well, just as we saw glucosamine. And galactosamine is also one of these amino sugars that you find in some glycoprotein hormones, like the follicle stimulating hormone and luteinizing hormones. These are hormones that we see in some reproductive development in puberty. You see these hormones when you are doing some endocrine system. I don't know if along your course you do something with the endocrine system, some fundamentals of endocrinology. You will come across these some of these hormones, which are peptide hormones. This particularly is a glycoprotein hormone, which means it's a combination of galactose, which is a monosaccharide or carbohydrate, with some proteins bonded together in that complex, giving you these hormones. But another aspect you can also talk about with galactosamine is that it has a hepatotoxic effect. That is, it can have a toxic effect on the liver. So in the laboratory, a lot of times people who are doing some animal studies, particularly with some rats or some mice, they induce say the condition of hepatitis. It can be modeled by injecting galactosamine into these rats. So because it induces some form of inflammatory reaction in the liver of these organisms. what happens is that you see when you take cells, we refer to ATP, adenosine triphosphate, as a primary source of energy for many of these cells. But in hepatocytes, that is in liver cells, you also have uh, uridine triphosphate. So that is to say that, in place of adenosine, you rather have, or let's say in place of adenine, that maybe you can quickly, because you did some of these in secondary school where you saw adenine, guanine, uh, as purines, and you saw cytosine, thymine, uracil. You saw this. So if I say uridine triphosphate, it means that in place of adenosine, you have in place of adenine, you have uracil. So in liver cells, you have this uridine triphosphate, which is a source of energy. It's a form of cellular energy for the liver cells, the hepatocytes. But what galactosamine does is that uh, it goes to deplete that energy store. That is, it goes and binds to uh, adenosine diphosphate, which means it will cause some dephosphorylation of adenosine of uridine triphosphate. In other words, what it goes to do is it goes to deplete the energy and the energy store inside the liver. And if that happens, it means that metabolic processes in the liver cells will be disrupted. And that further results into inflammatory reactions, causing some inflammation in the liver. That way, if it's used as an agent to induce hepatitis, it's understood. It goes to induce hepatitis in the liver of these experimental animals, and then that is used to further study this condition. Then we have monosaccharide. So we see mannose on the left, and monosaccharide compared to it, you see in place of a hydroxyl group, again on carbon number 2, you have the amine group there. So that is monosaccharide. No, monosaccharide by itself, not much to say about it, but except that it's a precursor for the synthesis of N-acetyl monosaccharide. So it means that on this amine group, you have an acetyl group coming to bind onto it. But even that N-acetyl monosaccharide itself only goes into the synthesis of other monosaccharide derivatives. So it goes into different reaction pathways. But itself, monosaccharide and N-acetyl monosaccharide themselves, not much to say about them, except that they serve as precursors for other modifications. We have also N-acetyl neuraminic acid. So here, this looks more complex. Different things are happening because you can see that on carbon number 1, this time around, it is not the hydroxyl group which is being replaced, but rather you have in place of hydrogen there, a carboxylic acid group is on the anomeric carbon. Then we also see on carbon number 4, some N-acetyl group, an N-acetyl group on carbon number 4. Then on carbon Number 5, we now see, or let's see, no, that is actually not carbon number, though it's a replacement of carbon number 6, where so there's the carbon number 6, which was a CH2OH. In place of it, we now have this whole thing, R, which is, if you take a critical look at it, this is a residue of glycerol. So you have this R. So it's a more complex molecule, and acetylnuraminic acid, which is a component of some glycolipids, which we refer to as gangliosides. So you have these N-acetylnuraminic acid, which will form a complex with some other lipids. And this you find in the membrane of many nerve cells. Sometimes you find it like sort of if you find it in things like sphingomyelin, for instance, that is you have some lipid, which is sphingosine, which is bound onto some of these, and they form some what we refer to as myelin sheath, giving protection to these nerve cells. You find N-acetylnuraminic acid as a component of some of these gangliosides. So you find them in the brain. So they are in the biomembranes of some brain cells, some nervous cells of the nervous system, neurons. So that's where you find some of these N-acetylnuraminic acid. Thompson Joy says that, sir, please, can you be uploading the recorded lectures on LMS for us? All right. So, well, the lectures actually, when I share the link with you after we finish, they are there. They are online that you can always watch. You can even download them. And the link I share with you is also another ATU, platform system there. It's just that it's double work to be downloading from here and then going there to go and upload it. Another thing with the LMS is that you can't upload a video that is above a certain size. It gets difficult. So usually it's advised that you put the link there. So the link will still bring you back to the link I share with you. That's why you don't find that I'm uploading things on the LMS. Also because a lot of times with experience, I realize that some students do not have access to LMS when they have not paid all their fees, so that every time you find the lecturer doing double work, you put the thing there on LMS, but not all the students can have access to it, which means you must find another way to let all the students have access to it. Because in any case, the student will come to pay school fees and write exams. And the student must study before he writes the exam. So that's why you find that on the LMS, I'm really not putting anything over there. But once I give you the link, you can always have access to it. The link usually on the Microsoft Teams is active for 90 days. Or no, I think 120 days. So that definitely covers the whole semester. Then later, when I have time, I also download it and I upload it onto my YouTube channel so that it can be there all the time. Not that once the 120 days is over, it means it's off. Because sometimes some students later, they want to just go look at it so that it will always be there. And the same thing, if it is on LMS, that one too, after the academic year, everything is wiped off, and then you have to do it all over again. So it's sort of stressful. So Joyce, I hope you understand why I'm not putting it over there. All right. Okay. Channel. Okay, my YouTube channel. Well, I think I shared it before, but I can share it again. If you go, I think this is my handle. It's my handle. Let me just type it in for you so that, but you remove the quotation marks. I have to put quotation marks there because when I put at it, as if I want to write to somebody there. So that is my handle on YouTube. When you go there, there will be some videos over there. But I think so far the lectures we've been having, I have not put anything there yet. Over the semester when there is so much of activity, I don't have the time to be uploading there. But later, at the end of the semester, I look through and then I start uploading them. But for now, you can still have access to these videos. They are still active for 120 days. The links that I share right after the lecture, you can have access to them all the time. And I set it in such a way that you can download it. Okay. Then we looking at also what we refer to as deoxy sugars. Deoxy sugars. First one we're looking at is fructose. So here too, you can see in place of the hydroxyl group, or you can, so this time around, you see the carbon number 6, which was CH2OH, that hydroxyl group has been removed, and you now have CH3. So that is why it's being referred to as deoxy. This derivative you'll find in abundance in the cell walls of the cells of seaweed. So that's it. That's where the name comes from, because in Latin, seaweed is called fructose. And so that the name, because it is found in abundance in seaweed, that is why it's being given the name fructose. So that is a deoxy sugar. You can see the hydroxyl group is removed from carbon number 6 over there. Now you're left with just CH3. You also have what we refer to as rhamnose, also a deoxy sugar. It's not the same as fructose. If you look at the orientation of the various groups that are on it, you will notice the difference there. Then we see this is also isolated from a plant, like what also called rhamnose. So we have some rhamnose species from which this sugar is isolated from. It's also found in some microalgae, and it's a major component of some bacteria, the Mycobacterium species. That should give you an idea, the bacteria that causes tuberculosis, Mycobacterium tuberculosis. So you can find rhamnose in abundance in the cell membrane of this Mycobacterium. And so some biochemical tests for the determination of the sugar is used to determine infections of Mycobacterium. All right. Then the very last one we can take a look at is, from the beginning, we talk about the glucose 6-phosphate that is in a glycolytic pathway in the breakdown of glucose. Now, along that glycolytic pathway, you come across fructose 1,6-biphosphate. Remember, the glucose molecule is to be broken down into 2, and the 2 will have 3 carbons each in them, and each of them will be phosphorylated. So you see that you are now having phosphate group on the first carbon and phosphate group on the last carbon. So that now when the molecule is broken into two, you can have phosphate group on each of them. So the fructose 1,6-biphosphate is an intermediate that you come across in the metabolism of carbohydrates. So that when you see it, you can quickly relate to it that, oh, this is a modification or is a derivative of monosaccharide. So on this note, we will end here. We have seen enough structures today, so we can go and assimilate it before we move on to the next, where we start to form complex big structures, oligosaccharides, polysaccharides, and even forming some polymers with some of these modifications that we have looked at. So I'll open the floor now for any questions, comments, additions, anything that you would like to say. All right, I see Doche Linda Ama. Your hand is up. You can unmute yourself and speak. Okay, so Emmanuel Dacha says the glucuronate is doing awesome work in our liver. All right. And glucuronic acid, yes, is doing some awesome work in the liver. I like that you relate to that very quickly and just practicalize it that way. That is doing some awesome work there in the liver. Yes. All right. So Doche Linda Ama, I thought your hand was up. You can unmute yourself and speak.